Hello friends, welcome to Medicine Hub 101. Hope you are safe and doing well. And today I just upload the video that is regarding the pharmacology and it's very important, the topic on the best of the pharmacology. And today we'll talk about the drugs for Alzheimer's disease. The drugs for the Alzheimer's disease. Okay, let's start the topic. So the topic is what? The drugs for Alzheimer's disease. Before going into details, I want to say you guys what is actually the Alzheimer's disease. Okay, so basically, that is a progressive neural degenerative disorders. It's a progressive neural degenerative disorders. So we can say what, first of all, on first point, what it's a progressive neural degenerative disease or disorder, whatever you can say, disorders. I'm writing a disorders. Okay guys, and what the pathophysiology of this disease, or the pathophysiology of the Alzheimer's disease is not completely known till today, but there is some, like, it's the pathophysiology is quite complex here. Okay, so there are some hypotheses regarding this disease. I'll just go through the hypotheses. There are basically the three hypotheses here, and I'll go through the hypothesis step by step. Okay, so what the pathophysiology of this, the pathophysiology, you can say what the pathophysiology of it means take it as Alzheimer's disease here. Okay guys, it pathophysiology of it is quite complex. It's quite complex and not completely understood. I can say not completely understood. Okay guys, now what you have to know here, that there are the few hypotheses by which you can explain the cause of the Alzheimer's disease. Okay, so let's come on the hypothesis step by step. There are basically the three hypotheses. Okay, so we can say what there are few hypotheses. There are few hypotheses by which we can explain that try to explain the cause of it, the cause of Alzheimer's disease. So basically we will just talk about what the first hypothesis. There is the most popular hypothesis. Okay, there is the cholinergic hypothesis. The most important hypothesis in this case, there is the cholinergic hypothesis. So we can say what the most important hypothesis is cholinergic hypothesis. Cholinergic hypothesis, guys. What about the cholinergic? Now you have to know what is actually the cholinergic hypothesis here. So this hypothesis states that what it happens due to the loss of the, this disease happens why? Due to the loss of the central cholinergic neurons. Due to the what? Loss of the central cholinergic neurons. So this hypothesis states that this disease occurs due to loss of central due to loss of cholinergic neurons, central cholinergic due to loss of the cholinergic neurons. Okay, and what? Whenever there will be the loss of the cholinergic neurons, then what? It will show what? Showing the deficiency of the neurotransmitter, that is acetylcholine, because we know there is the cholinergic neurons. Basically, they produce what the neurotransmitter, there is the acetylcholine. So whenever the loss of the central cholinergic neurons will be there, of course, definitely there will be what? The loss of there will be less number of what? Acetylcholine neurotransmitters. So we can write showing deficiency of, I write in brief form acetylcholine. Okay guys, take it as acetylcholine. Okay, acetylcholine. And what is acetylcholine? This neurotransmitter, it's a neurotransmitter. Of course, we all know it. And what is the function of this type of the neurotransmitter? Neurotransmitter that involves in memory and learning. So this was about what the cholinergic hypothesis. So basically what guys, the most important hypothesis is cholinergic hypothesis, which states that the disease occurs due to the loss of the central cholinergic neurons. This disease occurs due to the loss of the central cholinergic neurons, and they're showing that what? Deficiency of acetylcholine neurotransmitters. And basically we all know it, that's the acetylcholine neurotransmitter is basically involved in what? Memory and learning. So of course whenever there will be loss or the dysfunction of the acetylcholine, then what? There will be some dysfunction within the memory and within the learning purpose. So what basically happens in case of the Alzheimer's disease. This is one of the most important hypotheses regarding the Alzheimer's disease, guys. And now the next hypothesis that's also very, very important, that is called amyloid hypothesis. Amyloid hypothesis is also very, very important. Now this is the amyloid hypothesis here. So which states that, so this is the basically amyloid is basically what? On type of the misfolded protein, guys, on type of this misfolded protein. Okay, and that will be present within the extracellular fluid in between the brain cells. Okay, in between the brain cells. So what can we say from the amyloid hypothesis here, which states that it occurs, it means the Alzheimer's disease. Okay, it occurs due to accumulation of abnormal, or you can abnormally folded, or you can write misfolded beta amyloid protein. Beta amyloid protein. And what basically what beta amyloid is, what guys? Beta amyloid, this protein, we can say beta amyloid is we can say metabolic waste here. So beta amyloid is a metabolic waste product and it is present in the fluid in between brain cells, in between brain cells, guys. So basically what? So the amyloid hypothesis, this regards what? This states that that it occurs due to the accumulation of misfolded beta amyloid proteins, and the beta amyloid itself, it's a what? It's a metabolic waste product that is present in the fluid in between the brain cells. and what actually the problem is there. Okay, now I have to see it here. There guys, there's these beta amyloid proteins. So first of all, just assume it like this. I'm just focusing here. First of all, this like what? There will be like something like this. First of all, consider cells, and in between them what? In between them, this first of all, this is the neuron cells. This is the neural cells also, and in between them what? There is accumulation in the liquid, and within the liquid there is the accumulation of misfolded beta protein. This is the misfolded beta protein. Now this misfolded beta protein, they will combine, they will get together, all the beta amyloid proteins will get together and they will form a clamp, like the clamp. Okay, they will form a clamp. So beta amyloid proteins will get combined together, to form beta amyloid clamp. And for this beta amyloid clamp, they will get together and they will form what? There is a plaque. The beta amyloid plaque. Okay guys, this will form beta amyloid plaque here. Now what actually is occurring here? What actually we are seeing here? That this plaque, what you are getting at last, the beta amyloid plaque. This plaque, this plaque will induces what? The neural informations. It will just induces to interrupt the neural informations and disrupt the communication in the biochemical changes. Okay, we can say what? It will interrupt neural information in between the neuron cell. First of all, it's a neuron cell. Just assume it, okay? And here is one more neuron cells. And their clamp will get together here and they will inhibit the information to go from one neuron to another neuron. Okay, so they interrupt the what? Neural informations and what? And disrupt the communication. By this they will what? Disrupt the communication. You can write disrupt the communication. Okay guys, hope you understood. Now there is one more hypothesis that is called a tau hypothesis. Before starting the video, I just said what? There are basically the three hypotheses. The most important, that is what? The most important, that is the cholinergic hypothesis. Okay, this is the cholinergic hypothesis, and then we got what? The amyloid hypothesis. Now guys, last hypothesis that we will talk about what? The tau hypothesis. Tau is also itself a protein. Okay, itself also a protein. So you can write what? Tau hypothesis here. So tau. Now what is tau hypothesis? Which states that this Alzheimer's disease occurs. Due to accumulation of abnormal tau proteins. It's like similarly almost like like what it is, like the beta amyloid proteins. So which states that Alzheimer's disease occurs due to accumulation of abnormal tau protein, which form a triangle in between nerve cells. Okay, so what is actually this thing is here? Let's have a look on the diagram, then you can better understand. Okay, guys. First of all, there is a neuron. Still, what do you know? The neurons are here. Just assume like you are environment what. And first of all, there is another neurons. And in between them, a triangular shape. In between them, a triangular shape. This is like the what? The triangular shape. What? Formation of tau proteins will be formed in between the neurons. Okay, but in healthy persons, in case of healthy persons, the tau protein is having too much functions. It is having a crucial important role in case of the formation of microtubules. Okay, so in healthy, if I write what in healthy, brain, tau proteins help to form microtubules. Which helps in transport of nutrients and informations throughout the neurons. Set. So in a healthy person, in healthy brain, guys, the tau proteins help to form the microtubule. First of all, they will form the microtubules first of all, and this microtubule will help to transport the different type of the nutrients that is required for brain cells, and the information, of course, throughout the neurons. It will help. But whenever it will get accumulated by misfolded of its structure, or we can say the abnormally formed what the tau proteins, whenever will be get accumulated, at that time what will happen? So you can write what? By accumulation of its abnormal form. By accumulation of its abnormal form, what? Interrupts, we can say interrupts its biochemical communication in neurons. Interrupts the biochemical communication in between the neurons and nerve cells, whatever you can see. In neurons, simply I am writing in neurons. Okay guys, this was all about the hypothesis. Regarding the hypothesis, what the tau hypothesis, and I said about the the amyloid hypothesis, and at first I said what? That's the cholinergic hypothesis. So basically it's the progress, I said, the neural disorders, of course you know it. And in this disease we see the person, they are suffering from their, the loss of memory, and they are also to have what, they cannot learn properly anything. So this is basically the abnormalities in these patients. Now guys, we will talk about the drugs. Of course there should be some drugs. The scientists have tried to form the drugs against the Alzheimer's disease, but they didn't get that much the successful trials, but yet it is given the patients by which they can get a little bit curity. Okay guys, now we will talk about the Alzheimer's drugs. Alzheimer's drugs. So basically we can divide the Alzheimer's drugs in two types. One is, we can categorize it as the cholinesterase inhibitors. Cholinesterase inhibitor. Another, NMDA receptor antagonist. Receptor antagonist. Okay. Now this cholinergic, there is a cholinesterase inhibitor. Let's have a look here. It is in the presynaptic membrane, and we are having what type of this thing here? The postsynaptic membrane here, okay? And of course we know that what the acetylcholine, the acetyl-CoA, and from outside by food we can have the choline, will ultimately form what? Acetylcholine. ACH means take it as acetylcholine. Now there is an enzyme that is called CAT. That's called CAT, and this CAT enzyme will help to form the acetyl-CoA and choline will get combined together, and by the help of this enzyme, these two products will get converted into acetylcholine as the form of neurotransmitter. Now what is it? Choline acetyl transferase. We can write choline acetyl antiphosphorus transferase. So this was the enzyme which is helping to form what, acetylcholine from acetyl-CoA and choline. Okay. Now what? Of course this acetylcholine will just come from the presynaptic membrane. They will secrete the products. Okay. They will secrete the products, and of course there are some receptors. There should be some receptors for what? The acetylcholine, and those are the receptors. They will just come here and will act. Okay, and they will give the intestinal signals by which they can work their function here. Okay, now under normal condition in brain, what happens? Okay, just write it here. Under normal condition in brain, guys, there are some enzymes. Okay, whenever the acetylcholine is acting here, after their actions, there are some enzymes. Okay, these enzymes, what? This is the name of the enzyme is called acetylcholine esterase. And this enzyme will do what? This enzyme will take the acetylcholine, and it will break the acetylcholine into forms. One is what? Acetyl-CoA, acetate, we can say acetate. Another is choline form. So there are enzymes in the postsynaptic membrane. What the name of the enzyme is acetylcholine esterase, and the acetylcholine esterase will do what? The function, it will break the acetylcholine into two forms. One form is acetate, another form is choline form. Okay guys, and there is also one more enzymes that is called butyryl cholinesterase. There's the butyryl choline acetylcholine storage. And these enzymes also do what? This enzyme also can do the same thing here. It will take the acetylcholine and it will break the acetylcholine in two forms. One form is acetate, another is choline. So it happens when under normal conditions in brain. Now guys, so one thing here that what? Let's take it in response. Okay. Now the main thing you have to understand what if the amount of the acetylcholine is decreased. Why? Because in Alzheimer's disease we saw that there is the loss of the central cholinergic neurons. So if there is loss of the central cholinergic neurons here, then what? There will be of course there is less amount of what acetylcholine, and if there is less amount of acetylcholine, so what? We will not get that much acetylcholine here. So we have to make what? We have to apply such drugs which will not what? Destroy the acetylcholine here. Okay, that will not destroy the acetylcholine here. Then we can have the more amount of the acetylcholine to work their main role there. Okay, now there are some drugs by which we can inhibit the function of these enzymes and of course these enzymes also, and those of the drugs should be applied here. Then after applying those type of the drugs, they will inhibit this type of the enzymes. Then we can have more amount of the acetylcholine and they can work their main role there. Okay, but if we don't apply this type of drugs, what will happen? This one, as you know that there is like loss of the central cholinergic neurons, so there will be less acetylcholine, and after that if the little amount of the acetylcholine comes here, that will be also get degraded into by what? The acetylcholine storage and the butyrylcholine storage into what? Acetylcholine. There will be no sufficient amount of the acetylcholine in the postsynaptic membrane to work their main role there. Okay, so what we have to apply such drugs by which we can inhibit the function of this enzyme, the acetylcholine storage, as well as for butyrylcholine storage here. So what are the drugs? We have to know the name of the drugs here, guys. So basically we use what type of the drugs here? See, so you can see by inhibiting these drugs will increase amount of acetylcholine and their duration of action. Of course you know it that whenever the amount of the acetylcholine will be sufficient here, they can work their function better. And what? Of course what will happen? The amount of by applying these drugs, because they will not let the acetylcholine storage and the other enzymes to break down the acetylcholine, so amount of the acetylcholine will also get increased there. So what type of drugs we are using here? Donepezil. You can use the drugs here. Donepezil. Then we can use rivastigmine. Then we can use galantamine. Okay guys, this type of the drugs we can use in case of Alzheimer's disease, by which we can do what? We can inhibit the function of this type of the enzymes. But here one thing you have to remember, there is the drugs. Which drugs? Rivastigmine. This drug is having the capacity to inhibit both the enzymes, these enzymes as well as these enzymes. It will not let do it. Okay. So the rivastigmine is such drugs which is having the capacity to stop the function of the both type of the enzymes, that is here the acetylcholine storage and what butyrylcholine storage, and they will not let those enzymes to break down the acetylcholine in their products. Now another here, there is NMDA receptors. That is very very important. Of course the antagonist. What is NMDA receptor? You have to know here first. Then we will go through step by step. So guys, I am repeating here that what there are some drugs which the drugs are here. By inhibiting these drugs, will increase the amount of the acetylcholine and the duration of action. And what are the drugs? The donepezil, the rivastigmine, and of course there is galantamine. and only the revisiting minus drug is having such capacity which can inhibit the function of the both enzymes, that is for acetylcholine storage as well as for butyrylcholine storage here. Okay, now we will talk about the NMDA receptors antagonist. Antagonist means against, you know it only, right? Now see it here, same thing we'll just do. Okay guys, first suppose the acetylcholine is here within the vesicles, and of course we know that, just assume there is like NMD receptors, assume that's NMDA receptors. Now whenever the stimulus will come by the action potential, changing in action potential, of course we know it also. The transmissions, all the things will come here, you know it only. What these things, all the things will, that due to the change in action potential, what will happen, the stimulus will come from the presynaptic membrane to the postsynaptic membrane. Now guys, what will happen here, there are some, you know, there's like glutamate. One thing is very important here, what is the function, and there's also that is having the glutamate receptors. There is the, on top of the receptor here, what is that, is the glutamate receptor here. The research suggests that, what the researchers, they suggest that what, that the beta accumulations protein leads to increase extrasynaptic there's like the beta amyloid proteins. What will happen then the beta amyloid protein, whenever the amount of the beta amyloid proteins will get increased, then what, it will not let the get connected with the one neuron to another neurons. So research suggests that whenever the beta accumulating proteins, or you can say beta amyloid proteins, will increase, then what, it will leads to increase extrasynaptic glutamate level. By inhibiting glutamate uptake or releasing of glutamate from glial cells, from the glial cells. Now guys, what will happen, actually see it here, and there is like the glutamate receptors. Whenever the glutamates, first suppose if there is what, first suppose these are the glutamates here, and the glutamate will come here, first suppose take it as glutamate, and the glutamate is having receptors here, okay, and binding of the glutamate here, binding of the glutamate guys here to NMD receptor results in influx. Whenever it will get bind with it, at the time there will be what, influx of calcium ions within it, influx of calcium ion. That means the glutamate, whenever it will just get bind with what the there's like the NMDA receptors, at the time what will happen, there will be the opening of the calcium channels, and by which the extracellular calcium that will come within the within the prosynaptic membrane by the what influx process, which controls whenever this calcium ion will come here and get accumulated, it will control what controls the excitability of the muscles, excitability and synaptic transmission. You can see excitability and of course synaptic transmission guys. Okay, so when the accumulation level of abnormally get increase of the what glutamate here, so elevated overstimulation, so we can say what the overstimulation at the time, whenever the elevated amount of the what glutamate will be here, whenever what for suppose the beta proteins, the beta accumulated proteins will not let the glutamate to get bind with the NMD receptors. So what will happen, the glutamate will come here, then glutamate, more amount of the glutamate will come here, and they will give a stress, too much great stimulus, and that what after that there will be influx of the too much calcium ion within it. So we can say, so when the accumulate, when what will happen, the glutamate level gets abnormally elevated, overstimulations will be happen. Of course, overstimulation within the NMD receptors, there will be too much what accumulation of the glutamate, and that will trigger the NMD receptors. So what it is here, that the research suggests that the beta accumulating proteins leads to increase the extra synaptic accumulation level by inhibiting the glutamate uptake. So if it is not taken by anything, then what will happen, the accumulation will be too much, and it will do what, it will give the too much trigger stimulation within the NMD receptor. As a result, there will be too much influx of the calcium ion within it. So whenever too much calcium ion will just get entered through the influx process, at the time the cell get ruptured, there may be the cell destructions, or maybe what, the cell will get die. Okay, this is the process here by which the calcium ion, too much influx of the calcium ion can be caused of the death of the cells here. Now we can use one type of the NMDA antagonist here. NMDA receptors. Antagonist. That is our memantine. We can use the memantine here. Yes, we can use memantine here.